their symptoms started declining as they went. The um yeah. My last phase was machine learning. This is where I took my three different algorithms, random forest, XGBoost, and logistic regression, and I compared them on an AUC curve and found out which one predicts the best. Logistic regression won with an AUC of 0.659 because logistic regression works best with binary values like yes or no. And my data set was full of zeros and ones, which is binary. And taking this AUC of 0.659, it means that the model randomly selected Parkinson's patient above a healthy patient is 65.9% of the time. But looking at this AUC, it also told me one more thing. It told me that my model is definitely not ready to go out yet because it's recommended for your AUC to be zero point 0.9 or something like that for it to actually be able to predict like the best and actually go out. So this told me that I definitely also have more stuff to improve on. For my conclusion and real world impact, I found out that my hypothesis, specific REM sleep behaviors such as sleep disturbance, restless leg syndrome, and dream fighting, are highly accurate warnings for Parkinson's disease. And my key findings were 14 sleep features showed significant links to Parkinson's. Sleep disturbance doubles Parkinson's risks. Longitudinal tracking detected divergence between converters and healthy controls up to 9 years before diagnosis. And logistic regression, my logistic regression model received an 8 UC of 0.659 using non-invasive behavioral data, resulting as the best AI predictor we tested. For my future research, my limitation mainly was my data set because it just had yes and no data. It didn't have actual things that happened. And another thing I could add in, which I found later, was the age and the family history of the patient. My next steps are having little trackers on each person. So if they wore like little sleep rings, smartwatches, or whatever, then I could actually track it and I could collect my own data and I could collect whatever I wanted, not just what PPMI had collected for me. Not for me, but had collected. Um, and yeah, pretty much. Thank you. I think you're on mute.
Yeah, I am. Thank you. So we definitely have some time for questions. Thank you so much for sharing your project. So judges, we have uh, we have Dr. Priyanka, we have Miss Allen, and we have Dr. Dusty. So what what do we have as questions for Ishita?
Ashita, go back to the machine learning slide. Okay, explain this. What is an AUC?
An AUC is like a curve, and I'm like still a little bit, I need to learn about that a little more. But
Yeah,
an
help me
AUC,
understand it.
an
Uh-huh.
AUC is like a curve, and it's it's like model testing. I think that's how it works. But
Mm. What did you use like on your device? What are you looking at when you are doing this? You're using a program to make this?
yeah, what I did was I split my data into like two different sections, and then I like trained the program with half of it, and then I tested it with the other half to see how
Oh, okay.
predictive it was.
And then can you go back? Okay, got that. Can you go back to, I don't know, maybe 3 slides? One more? Yeah. Okay. Do you know what the 14 features were? Because you said that you gathered 12 symptoms to monitor. I think you said that in the previous slide that you gathered 12 symptoms to monitor.
Yeah, no, that might have been a typo.
Okay, and then in the results there, it says 14 out of 20 features. No, in the slide that we were on. Yeah, well, even here, it says 14 sleep features. So so if you gathered 12 symptoms to monitor,
I think it was 14.
is it 14? Okay, because I can't track this. Okay, that makes sense. That makes sense. Okay, good, good, good. I think that's all the questions I have for right now. I'll probably think of another one in a minute.
Hey, Shita, hi. On the same topic of 14 features, were all the features rated similarly, or did any of the features were given better, like more points versus the other features? Like out of the 14, how was the spread?
Um, most of like there's wait, if I go back here. Basically, there's a base, like this is the odds ratio.
Mm-hmm.
And basically, I just chose the features that had more than a 1.0 odds ratio because 1.0 is like the baseline. It's just
Mm-hmm.
like, um, and if odds ratio like is less than one, then it's related with lower odds of the actual Parkinson's disease being there. So I just chose everything that had 1.0 and more.
Okay.
Okay, I have another question. What's dream fighting?
Dream fight. It's a symptom. It's like in your dreams you can like you physically act out and you I guess you just start fighting.
Hmm. So there's going to be a physical response, or you're going to do something physical, like
Yeah.
your body's going to move. Okay, okay.
All of them are physical because they're from RBD. So
Right, right.
So then, okay, won't that also be a part of sleep disturbance? Like your sleep is getting disturbed? Anyhow,
yeah.
like, so how do you, like, you're separating this dream fighting from within sleep disturbance then? You know what I mean? Like,
Yeah, I see what you mean. Because would dream fighting count as sleep disturbance? Yeah. I'm not exactly sure how PPMI did it, but I was
okay,
assuming that sleep disturbance was like you wake up in your sleep not physically acting out as much, but you wait that your sleep's not continuous. That was my thing that I thought, but
okay.
Hi, I had a question.
yeah, go ahead.
I was just wondering, so you mentioned using the ring data, uh, and you wanted to use more than just the data that initially you presented.
Mhm.
Would you like to elaborate on what other data you would use?
Yeah, so PPMI has like demographics for each patient. So they have age, they have family history, as I remember, and they had a few other things like how like what years they visited exactly. So maybe using those, I could add more to my data and then get out more conclusions like people within this age have a higher likelihood or something like that.
So you would increase your data set based on publicly available information or HIPAA protected information? I guess that's kind of the question I was going towards.
PPMI's publicly available data set.
Thank
Yeah.
Well, we are at our 15-minute mark right now. Um, so that should be enough for questioning to keep it the same for all competitors. So thank you so much, Ashita Jane. Uh, we will get back to you with the results, not today, with the actual direct feedback, but we will have everything for the award ceremony results later in the day.
Okay, thank you.
Yeah, bye. Awesome. All right, good work, judges. So we are, just wanted to let you know too that we are officially live stream um, at the moment too. So you might have gotten an announcement or something. So apologies for doing that. I should have apologized to Ashita too. I probably threw her for a loop, but anyway, we are doing that. So how was your first judging experience with Research Innovation Expo with STEAM and HS?
She was surprisingly eloquent and confident. That was awesome.
Yeah, we have
Good
some
project.
really good competitors, and we're looking forward to our next for sure. We have a break right now because we had a group a group project that they ended up canceling their slot. So we'll have our next participant come in at 10:15. So if anyone needs a break, go for it. What I'm going to do right now is I'm going to share our video that we have created. If you want to see a little bit more about it, feel free to watch. If not, you know, ignore it. I'm going to go through and I'm going to make the rubrics for the rest of the individuals from today so we can access that. All right. Those are all really good questions that everybody was asking, so I really appreciate that. Elizabeth, thank you for sending the NOAA Sea Grant
Isn't that
event.
cool? Isn't that cool?
Yeah,
I'm so excited. I'm like, please clean up the ocean. This is like, all right, like let's get going. I've shouted that out quite a bit this week to my coworkers. I'm pretty sure they're like, take it down 10 notches. So for the other judges, I'm happy if she wants to send it, if Sarah wants to send it on to you, it's fine. It's just next week they have a guest speaker who's talking about how to use AI to get autonomous vehicles to pick up pop.
yeah, and sorry, sorry the video was playing that thing. The and I did have a question. So for that, is it virtual as well, or is it all in person?
It's virtual. It is free. So like they
Oh,
just
cool.
have so many slots for guests that come in and then you can ask them questions. It's really cool. And they usually have like teacher connector pieces with it so that you do something with the knowledge and go forward with it. But yeah, I think that's pretty cool. They're doing some interesting stuff. I'm glad they got the grant. I really am very curious. There's a company out of Alameda that does autonomous drones, but they just do data collection. They're not picking up the plastic. So I want to see exactly how they're implementing it.
Yeah, it's going to be really interesting.
Yeah, I hope so. Do I need to, is there like a form you'd mentioned a form and a folder? Do I need to,
Oh,
thank you.
oh yeah. Let me send that to you. Sorry, I thought you had it. Um, Priyanka, while I'm getting that to Elizabeth, so there the if you go to the link that will take you to the folder, and then Priyanka, if you are able to stay to judge for the next one at 10:15, that'd be great. If not, I understand.
I can do
Is that
one more.
okay? Yeah, it's just a bummer. Like we had like someone cancel a no-show, right? Um,
I
I'd love
get
for
it.
you to see.
Happens.
Yeah, I'd love for you to see another competitor. These students are super, impressive and interesting. So
Yeah, sure.
awesome. Thank you. I really appreciate that. And here I can, um, while we're waiting, I'll share this video that we were kind of hearing before. So this is one of our other students. So this student's not competing here today. Um,
literally with this project. At the
but she's
beginning, it
gone
was
through this
complete
process
confusion. I
that
had
these
no
students
idea
have.
what I was doing or what to expect from this program or from myself. I quickly realized that the project can stem from simple everyday objects such as stress-relieving toys. I've probably seen them around in offices and schools, but I realized as I gathered sources that there actually isn't a lot of research backing their credibility. I was surprised given how much we take for granted that the stress toys do work, and organizations and companies hand them out to help in the workforce. This showed me that I can't always take everything for granted and that you should always be questioning your surroundings to find more answers, even to things or ideas that you think are certain. This project has allowed me to expand on skills such as presenting, writing, and researching. It has given me the confidence I need to continue researching and innovating past my high school years and into college. Thank you.
So I just messaged the student who's at 10:15, and we shall see. Ah, there you are. I just messaged you
Yeah,
here.
yeah, I see.
I'm
I
so glad you're here. We've had a couple people not show up, so I'm so glad that you're here. Okay, excellent. All right, dear. So we'll start in 5 minutes. All right. So if you want to share your screen, get a handle on that.
Yeah. Uh, sure. I can't really present it directly on slides because I'm not home right now. I'm out somewhere outside, so I'm relying off a hotspot currently. Now that it's a PDF. I'm sharing it off of.
Okay. Do you need me do you want to send it to me via WhatsApp and I can share it on the
I think you already have it. Uh, remember, yeah, it's not really loading up for me very well. It's like stuck on one spot.
Okay. All right, let's figure it out.
I could share off with this PDF. I have a PDF downloaded of it. When I put it, but when I put it
Oh, yeah.
into the form, I have downloaded that.
Okay, yeah, give that a try. See if it see if it works for you.
There. Yeah, it
Perfect.
works.
Yeah, it's showing up. That's wonderful. Thank you.
Yeah, I'm happy I downloaded it because if I didn't download it, I couldn't really access it well.
Oh, good. Well, I'm glad you downloaded it. That was good thinking. All right, judges. So we have files for Veer made.
So I could start now.
Yeah. If you can go ahead and start now. We will. Uh, let me go ahead and once you start, we'll have about 10 minutes for your presentation, and then would like to then have about 5 minutes for questions. So you'll have 15 minutes total. All right. Just to keep it the same for each competitor.
Okay, so this is my idea, Blue V, the next gen pool water tester by Veer. Research question background. Like I I when I was making this, I thought, how can I make a device that's low cost, reusable, and used to test residential pools accurately? Because I know, and my background is pool testing. If you own a residential pool, you have a few good options to choose from. You could either get a pool shop or get some test strips, which one they both cost enough money as is, and the amount of test strips and the amount of time you have to go to a pool shop will be a lot. Like, because one bottle may contain like 20 or 30 of them, but the cost builds up over time, which is the only thing you don't want. And like if you use the best method out of those two with the test strips, as mentioned in background, like they have yes, they're accurate, but there's so much chemical waste coming from them that it's better to go to a pool shop. But again, you're spending more money there because you have to go there so often. Like on the right are like accurate or what comes from pool shop. Like these like over here there's like it's they're testing everything for water they most importantly there's chlorine, alkalinity, and pH hardness for water, which are like the most important things you need to know for testing pool water, because if one of them is too much, you can't swim in it. Currently using Arduino R4 with an analog pH sensor, analog turbulity sensor, and an analog TDS sensor. The TDS sensor means total dissolved solids, which is like out of parts per million. Like ppm, you see, like if you see water has 80 ppm, there's 80 parts for every million pieces, like pieces in it. So like 80 parts of plastic for every like, let's say for example, like for every million pieces, like they're in the ocean, right? The turbidity sensor like tests fogginess in water. And it's important for this because my idea isn't just you watch it. If you leave it in, you forget about it, you do work and you forget about it. The majority of people who own a house, they have work too. They because no, who own a pool, they have work too. Right? And like owning a pool isn't a cheap thing anyone can do. Let's say if you got it made, you spend money on just making the pool alone. So you have to work for that money. So you don't have time all the time just to stay there for 30 minutes. Like in those 30 minutes you could make more money so you could manage the pool better. So like, because I want to even forget about it, like, it should make sense like how foggy the water is as is. So if you know if it's too much or too little, like is it concerningly a lot of fog in the water? If it is, then it'll know because you're not there to tell it. You tell it to that. The analog pH sensor, I think it also senses temperature thing too. It senses pH as you know, right? Like for me, like this is the TDS sensor, the pH sensor, right? VDS pH, and this is a turbidity sensor. Actually, I also think this one also does voltage in the water, like how much volts the voltage of the water is. I've done testing for this stuff and getting it working. I know how it works, and you see here, it's like this is the board I'm using for it. This is currently what the diagram looks like, and this is what I have currently. It's like I still looking. It's like a wire mesh right now. Otherwise, mostly is working, right? At the everything everything works well as is, which is one thing I can say, which is good. I still have to I still have to get the one value dialed in, and also get it boxed up because I said earlier, I want to be you can forget about it and not have to worry about it all by the end of it. Which also means getting these wires shortened, which I've already started doing a little bit of it. But this is um this is the name of it, and this is where I got like where I'm basing off of. It's it's meant to monitor water quality, but for me, I'm using it to test water quality instead. This is I've been to I've been to places with like test strips. So let's say you're colorblind, for example. So someone says it's green, right? Let's say, for example, this one, like someone tells you it should be orange. If you're colorblind, you wouldn't know what orange is. It's just one small example, but otherwise, this is how it looks like if you go through. If you the different types of like the water qualities look like. Of course, it's based off of pool water, so you see it's not really optimal for swimming in. Using test strips, they're single use. It's convenient to use, but it's expire in a minute. So if you leave it in, take it out, but let's say you get a work call, 30 minutes and a minute later, the results are gone. So you have to spend them, you have to get another one. You got to take out another piece to test the water with, which is really inconvenient, right? There's storage requirement because you need to keep it in a cool, dry. You need to keep it in a place that's cool and dry. You can't keep it in a wet place because then it'll test the water that seeps into it. And you was wasted like $5 because the water got into it. It's affordability is a big factor in this because yes, it's affordable, but 50 bottles, but 20 bottles in, it starts to eat up on your expenses. Accessibility, as I mentioned earlier, if you have color blindness, and yes, it can work out, but let's say in these specific situations, right? Like the situation I mentioned earlier, you don't know if it'll work out. Right? You don't know, like you may be confused about it because it's a color, right? It goes by visual, like by visual appearance, and it may be altered for you. So it may make it very inconvenient for someone else. Movie. one advantage one advantage Bluey has it's reusable. Yes, it'll cost more, but over time it'll over time it's just a one time spend. And if you need to fix it, you can, and since all these parts are commercially available, and all the, so you could just buy you could buy some wires. You plug in the wire that's damaged and you fixed it. It's also expensive because you'll go get a whole bunch of wires, but here you have a bunch of replacement wires now. If anything else goes wrong, so it's another one-time cost. It's convenient to use, right? Unlike the test strip, it'll be on a screen, and it'll be on a screen or something, right? There'll be a SD card for like the history of it, right? But the screen will make it so no matter if you're colorblind or not, you could see it and say, okay, it is safe to submit. It has too much coding or too loud. It's very clear. It's clearly in front of you and no colors indicated. It's just words and everyone understands words. It's like as I mentioned repairable earlier, it's accuracy and accessibility. I'm I mentioned I remember when I mentioned blindness effects. Within Bluey, as I mentioned earlier again, the color blindness will affect the way you see the results. Because the results are as what's shown on the screen, it is be accurate towards like the results will be accurate like the test strips and also it'll be more accessible to people because no matter if you have color blindness you can still see red, you can still see it. Right? You don't need anyone else to tell you what it is. It just you put in water, you take it out, and you read the results. Like affordability, as mentioned earlier, again, it's just a one-time cost. You buy it, you don't have to buy another one unless it's severely damaged. But if you take perfect care of it, it'll not get severely damaged. You could always repair it. So so on the test strips, you could just buy one and you don't need to buy it for like many years afterwards, possibly. Get in conclusion, this data will include pH hardness, chlorine concentration, and affinity. In my initial model, my goal is for accuracy within basic parameters. Like, is the pH enough? Is the hardness enough? Is the alkalinity enough? Right? Is it falling within a certain range? Because if you be sort of work machines, you know what tolerances are. And the tolerance for this, right? It doesn't fall within a tolerance. Because of course it'll be off. It's technology. The biggest flaw is also its greatest weakness. But like but just how off will it be? If it's too off, right, it'll not help you at all. But if it's off enough, right, you could still get results accurately and like you'll be in a position where you could always get results that are accurate enough for your pool. It, that's that's it. That's yeah. That's it for now. Whereas, thanks, thanks for this. Yeah.
Yeah, awesome. Thank you so much for sharing about your project. We really appreciate that, Veer. And we definitely have time for questions. So, yep, we definitely have time. So, judges, do we have questions for Veer?
Vera, for
No.
for the turbidity sensor, how does that work? Do you know?
Turbidity sensor thing. It's there's two prongs on the end of it. I have a general idea how it works. Like when you put the two prongs in water, it's like it just like measures like one thing it measures is the electrical flow throughout the water, right? So if there's too much in it, like that it could hurt someone that you wouldn't know of, like it'll figure it out on one end, like because like if you steal like forks and all, like how it actually goes through forks, it works. I think works something like that, where it's like there's two prongs because it has to flow through it in some sort of way. Like I know you talked about this thing because I remember how it looks like.
So, and then go to your diagram, the next slide.
Yeah.
No, other way. Yeah. So, okay, this is your diagram. We've got the pH and the turbidity and the total dissolved solids, and the idea is to package all of this up into like a flotation situation.
Not really. I wanted to sink in the water. It was like for example, I have the robot who like I could attach it to that and let it clean. So when it comes out, it's like if it floats, if the purpose of what I want just gets defeated. So it's like it's like air, like a box that's like airtight, so it can't, and it's like doesn't have enough buoyancy in it to float. That's my whole idea of it.
Okay. So you're saying that your idea is for it to sink to the bottom, or
I'm
that
sorry,
that's where you're
repeat
at
yourself.
with it?
I'm not
Oh, sorry. So your device will maybe sink to the bottom of the pool then?
it can't, it would be able to sink to bottom pool, but I would intend it for it to like something. Like let's say you attach a string to it, you could let it sink and then you could pull it out after like 5 minutes or when you get free next. Or you could attach it to a cleaner while you clean.
Okay. Thank you.
You're welcome. Any more questions?
Hi, can I ask a question about your pH reader?
Yeah, sure, go ahead.
I was just wondering, when it connects with an Arduino, is there any risk for where the wires are? Like, I guess it's really cool that you did that. I just was wondering, are the wires going to be exposed to the water?
Um, Hopefully not exposed to water. Because like if you take it'd be fine if the rubber sections or wires were exposed to water because it just stops the rubber stops the flow. But if the actual metal is exposed to wires, that's a real danger. It can short circuit the whole thing. It'll just be a whole mess. Otherwise, I don't intend for any wires to be exposed.
Okay, so it's just the bulb that's in the water and the Arduino itself is out of the water and the wires are going to be sealed, or I guess that's the part that I can't quite tell.
I am intended for like, the parts I need to be out the water. I'll take that much out the water, like outside the case. Right, that's all I'll do. But otherwise, I'll keep the rest within the case. That's my idea for it.
Oh, okay, so there's a waterproof case that encloses this. All right, thank
Yeah,
Hello, I have questions.
sure. Go ahead.
Yeah, if you had redesign your system using a low-cost microcontroller instead of ordinary UN hardware which you used, what change would be the required?
So repeat yourself. My the connection here isn't really very stable. So yeah, it didn't. It was a little um scratchy.
So
What
the question was if you had to redesign your system using a low-cost microcontroller instead of something ordinary like the specific thing that you're designing, what hardware do you think that you'd be using? Which
If there was a mic,
blockchain do you
okay,
think
if
you'd
there
be
was a micro,
using?
I don't know the. Well, if it was a microcontroller I was using instead of the system, I'd probably instead of having one main control area, I'd probably divide it into more pieces because it's less power, so it can't it can't power the whole thing by itself. So I'd probably like, let's say I get I get versions that need less power to run. I try using less wires in it, so I keep the flow like of electricity is like directly like no complications, or try to make the simplest design I can possible. So that'll be what I would do if I was only using microcontrollers. But
Okay. So what specific modifications would you make to improve the accuracy of your device to reach near the laboratory standards? Because you have mentioned the pH of that your values. So what it will be like to the level of the laboratory standards?
um, like what level of understand this? I don't really know what level to understand this, because like I because if you do a little bit of research within this, like you're gonna get a general idea of what it takes for it. So I would say it's not a lot to understand it, but if you really want to get it, it'll take more of understanding of how it works to really know what's happening in it.
Okay, okay, fine, fine.
All right. So I think that we are at our time. And yeah, so we are at our time. So let's move to, uh, I don't think we have our next student here yet, but let's just thank Veer. So Veer, nice job. Thank you so much for explaining your prototype that you're going through and developing. We really appreciate you, taking the time out of another competition that you're in to come and do this.
Yeah.
So really appreciate that. And so have a good day, and we'll get you the direct feedback, not today, but the results will come out today, if that makes sense. So
Okay.
you'll
That
hear
makes
you'll
sense.
hear about the results today, right?
Otherwise, otherwise thanks for this. Otherwise, yeah. Actually
Yeah.
done. Have a nice day then. Bye. Also,
Yeah.
good morning. Good morning. Good morning for you guys. And the time difference. Like it's like
Yeah, and yeah, and I think we have Likit who's in India, so it's wicked early or wicked late, for him.
it's around 10, 11 p.m. there.
Yeah, really late. So, all right. Well,
Otherwise,
have a
glad
good
to
one, Veer. Thank you so much.
okay. You're welcome. Yeah.
Bye.
All
All
right,
right.
Sarah, I'm gonna drop off too, but this was cool. Thank you and happy
Yeah.
judging for the rest of the judges.
Yeah, thank you so much, Priyanka. I really appreciate it. And if you could, before you run off to the museum and have
Yes,
a wonderful day, if you could just fill out those
yes,
forms, I would really appreciate that too.
absolutely.
So,
Yeah, I'll do that right away. All right.
okay. All right, thank
Thanks.
you.
Bye.
Bye. And then Elizabeth, and I think Dusty, same, right?
Yeah, I've filled
Yeah,
out one. I'll fill out the other before, or I'm gonna get off and then I'll fill out the other.
yeah, that sounds good. Perfect.
Good
And
luck.
then, yeah, thank you. I really appreciate you doing this, and you and Elizabeth and Priyanka too were asking such good questions. So I really appreciate your time and support for these students.
Yeah, no
Thank you.
problem. Hope it goes well.
Yeah.
Thank you,
All right.
Sarah.
Yeah, thank you. Bye.
Bye.
All right. So, hello, our new judges. Hi. Um, I am Sarah Abood. You are the person who you were messaging with. I am in the comments here. I am going to be sharing a folder with you. And in this folder, actually, I probably shouldn't have said that. Hold on, let me delete that really quickly. Okay. So, there's that, and then there's that. Let's have, oh, yeah. So, Mr. Markle,
Yep.
Would it be okay? I'm trying to keep it kind of informal to keep the students from getting too um,
Hmm.
nervous. So I'm trying to use first names for the judges. I know that it's in no way to try to be um,
I go by
unpro
my first name.
unprofessional, right? I mean, I go by Sarah, right? So, yeah, right, and so I normally go by Professor Sarah or something like that, for students to be a
Uh,
little more like engaged with them. Anyway, so if I call you by something that seems like it's not your normal, please tell me and I'll change it. Same for you, Mr. Like, oh gosh, Likit. Is that? Please tell me how to pronounce your name.
Yeah, it's Likit itself. Yeah,
Likit
yeah.
itself. Okay, perfect. Okay, so Lakeith and Mr. Markle, we have uh, a couple of students that are coming up, and so in the folder that I shared with each of you, you will find that there are some files, right, and a schedule. And so we currently are we have a student who is already late, right, for this time around. But we also have Hashidi who is perfectly on time and ready for their presentation. So for her presentation. And so let's have Lakit, let's have you be judge one. So if you, for example, for Hashini's, you would go to the Judge One file and you would enter your feedback and comments there. And uh, respectively, right, Mr. Merkel, you would be doing the same for Judge Two. And we'll keep that number for the the individuals. So at 11, we're going to have Athmika as well. So are there any questions about the judging aspect or anything like that?
So in the Excel form, there are for student of two name. So basically, who will be taking first, like judge one and judge two?
Let me double check what. So for Hashini, if you go to that
There are two
file,
Excel forms in Harshini. So who will be filling the first form and who will be
oh,
filling the second form?
good question. Yes. So
Yeah.
the file that ends in Judge One, that will be you, and the one that
Okay.
ends in Judge Two, that will be Mr. Markle.
Yeah. Thank you.
Yeah, very good question. And then similarly, for the next student that we're going to be having is Athmika. Um, I am renaming that one right now, so it may not show up unless you refresh, but you will be filling out the Judge One for Athmika, and similarly, Mr. Marco will be doing the Judge Two. So if you
Yeah.
refresh,
Sure. Sure.
you'll see that one there as well.
Yeah. Okay.
Yeah, thank you. I really appreciate you asking that question because it,
Yeah.
it can be confusing. Yeah. All right. So
Yeah.
it's pretty straightforward. It's very similar judging at uh, mm, everywhere, right? We have Hashini, and Hashini is going to be sharing her slideshow presentation. Hashini, do you think that you could get that set up?
Yeah, okay.
Thank you so much. And then we'll start in 5 minutes, unless you really want to start earlier, which is okay with me. You'll still only have 15 minutes to go through and do your time slot to keep it the same for every participant.
Okay.
Perfect. And we will do around 10 minutes for presentation and then around 5 minutes for questions from judges.
Okay.
Perfect. So let me know if there's any help that you need for setup.
Do you see my screen?
I do. It's a little smaller than normal. It looks like more like a video or something, but
Oh,
it looks like
um,
a slightly different version.
okay.
Can you do the opposite of what you just did?
Like not share?
Oh, no, 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 sorry. It got small. Um,
I can see it. If you go to the next
yeah,
slide, you see how it looks with smaller font. Yeah, okay, that's pretty small.
yeah.
Okay.
Yeah, there's just a lot of black. Normally, you don't have as much black space on the side when you present. It might be because it's in a PDF form. That's my,
Okay. Let me stop sharing and reshare it.
okay, sounds good. This is the classic troubleshooting in a Zoom call uh, that everyone in corporate world deals with. So you are getting a skill set here.
Is that better? Could you see my screen more clearly?
This is lovely. Thank you.
Okay. Okay. Can I start now or do you want me to wait till?
Yeah,
Okay.
you can start. Yeah, thank you so much.
Okay. Hi, my name is Hashim Anod. Today I'll be talking about my project, enhancing the effectiveness of streptomycin using plant extracts against Staphylococcus epidermidis. To start off, antibiotic resistance is becoming a huge problem worldwide. Bacteria are evolving faster than we can develop new antibiotics, which makes infections harder to treat and easier to spread. Because of this, researchers are starting to look into alternative solutions, and one of the interesting idea is using natural plant compounds to boost the effectiveness of existing antibiotics instead of creating new ones entirely. For my project, I focused on 4 plant extracts: garlic, turmeric, neem, and tulsi. Each one of these has an antibacterial property. For example, garlic has something called allicin, which damages bacterial cell walls. Turmeric has curcumin, which interferes with bacterial enzymes. Neem slows down bacterial growth. And tulsi has eulogy, which inhibits bacterial growth as well. So I tested Staphylococcus epidermidis, which is mainly harmful skin bacteria, but it can form biofilms, making it resistant to antibiotics, and we can use it commonly to test antibacterial activity. So my research question is, can neem, turmeric, tulsi, garlic extracts enhance the antibacterial effectiveness of streptomycin against Staphylococcus epidermidis? And my hypothesis was that if Staphylococcus epidermidis is given to streptomycin with any one of the plant extracts, the zone of inhibition would be larger than with just the use of streptomycin. So my methodology was, first I put on the protective equipment and I made sure to sterilize all the work surfaces. Next, I prepared the plant extracts. I weighed about 0.5 grams of plant material. I crushed it with the micropestle and I added 4.5 milliliters of 70% ethanol to the plant material, and I let the extract sit on the windowsill for about 12 hours so the alcohol can evaporate and it will just be the microbial properties. Then I inoculated the plates. So basically, I swabbed it once, turned it for 90 degrees clockwise, swabbed it again, then turned it 45 degrees counterclockwise, and I swabbed it again. And then just to make sure, I just swabbed the outer rim of the circle so the colonies would fully form. Then I prepared the disc. I soaked sterile filter paper with 5 drops of plant extracts, and I prepared the plant disc as controls. Then I placed the disc in the center of the plates. I labeled, sealed, and incubated the plates for about 24 to 48 hours. After that time period, I made sure to put them in the freezer. This basically ensures that the plates don't grow again and I could look back at them for future studies. Then I measured the zone of inhibition. So basically, it's this clear circle around the disc, and basically, you would measure the diameter in millimeters. Then I recorded and analyzed my data. So as you can see here, on the left is a dot plot. So basically on the x-axis, I have my treatment groups. On the y, I have the zone of inhibition. So this is just showing each of the trials and what they produced. And you can kind of see visually the range of the trials. For example, turmeric has one trial which is 0 and one trial with 15 millimeter zone of inhibition, and it shows how much how effective the treatments are, and it makes it kind of predictable. Okay, is garlic going to be maybe 13 if I run trial number 6? And on the right side, I have 2 agar plates, and they clearly show the zone of inhibition. And this is garlic 4, trial 4 and garlic trial 5. And on the left, I have another graph. This is the 95% confidence intervals for the treatments. So they basically show the mean zone of inhibition, and they show kind of the range of the turmeric and streptomycin, because the range shows, okay, I'm 95% sure that it's going to be falling between maybe 3 millimeters to 16 millimeters for turmeric and streptomycin. On the right-hand side, I have another agar plate, and this is the Tulsi only. As you can see here, the colonies are really bunched up around this paper disc, and this shows that Tulsi actually helped the bacteria grow and didn't inhibit at all. This is another analysis. I did the t-test and the turkey hawk test, and this kind of shows that neem and garlic are proven analytically to effective against Staphylococcus epidermidis. So the results are that garlic and streptomycin had the highest mean zone of inhibition, which was 13.9. Neem and streptomycin had 11.9 millimeters. Turmeric and streptomycin had 9.2 millimeters, and Tulsi had no inhibition. I also tested plant extracts by themselves, but I only did one trial of them because I did not have enough plates. But the results are that neem had 24 millimeters, turmeric had 15, garlic had 13, and Tulsi had 0. And streptomycin alone by itself had no inhibition, which kind of shows that the antibiotic was already resistant to the bacteria. So garlic and neem have potential at synergy, and we could use them more often in developing future drugs. However, turmeric and streptomycin had a lot of variability. It was less consistent and it had lower effectiveness. So we wouldn't really use turmeric a lot. Perhaps a better extraction method would better allow it to have more consistency. Tulsi and streptomycin had no inhibition, and this could be because we used the wrong extraction method or we did not use enough of the plant. And some limitations were that we only had one bacterial strain and antibiotic tested. We also did this during the winter as well, and it was really hard to procure fresh plant material. So that could also kind of lead to reduced efficiency. And this isn't really generalizable because I only used one extraction method, and if you use various extraction methods, the data won't always be the same. It'll always have some degree of variability. So for future research, I would possibly test another antibiotic. One antibiotic I was actually looking into was ciprofloxacin, and another thing I would do is I would look at like what was the lowest amount of plant extract could I use so we can produce cheaper, better drugs. I would also actually quantify the synergy using the fractional inhibition concentration index, FICI. And I would also include more plates for the no extract and no antibiotic to confirm that inhibition was due to the treatments and not to the plant itself. And I would also look at how the treatment um, inhibited over time so that we could say that, oh, this can be used for long-term effects, or this is just a one-time thing. So these are my sources that I use, and yeah, that's it.
Thank you so much. We definitely have time for questions. So we have about a little more than 5 minutes for questions available. And then we will move to the next participant afterward. So, judges, what questions do we have for Hashini?
Ashini, can you go back one slide?
To the future research.
Yes, yes. Okay. What was the quantity of the plant extract that you used for your experiment? I might have missed that in the beginning.
So the quantity that I used was I did like a 70% extract and alcohol. I did not do 100% because I kind of figured out that based on previous trials that it got kind of stuck and kind of clunky. So we decided that 70% was the best one and it would work better with the alcohol as well.
Got it. Okay. And so you use 70% for each of the extracts, is that correct?
Yes.
Okay. All right. I think I do see in your future research that you plan on doing more concentrations. I'm just thinking about the what was it, Tulsi
Mm-hmm.
that actually inhibited the antibiotic. I was wondering if you were going to test that at a higher concentration, but I think I see that here.
For Tulsi, I would actually use a different extraction method because that didn't inhibit the bacteria. I would try to maybe go more water-based or perhaps use a different alcohol.
Okay. I was just thinking that it may have not been successful because of the concentration as well. So maybe
Yeah.
test both of those. Yeah. And another question I had is, um, I know that these extractions have their own antibiotic properties. Is there a reason why you chose these four?
You mean the plant extracts?
Correct. You know, out of all the extracts you could have chose, I know these ones, I know they have antibiotic properties, but so do others. So
Mm-hmm.
why these four? Do these four have, do you have like background information that shows that these already work with strep or, you know?
No, not really with streptomycin. I kind of chose these based on my cultural background because my parents use this a lot in dishes and they usually give it to me when I'm kind of sick or with the flu. So I always thought that these would kind of, stop the flu. So that's why I kind of carried that forward into that experiment because I actually wanted to see if it actually worked and wanted to quantify it.
I see, I see. You were empirically testing and questioning your parents. You took that to a new level,
Um,
right?
Well, I mean,
The-
I proved them right. So most of it was right, except
yeah,
Tulsi.
yeah. And that's just with this one, with that one antibiotic, correct? Yeah.
Yeah.
So, other questions from judges. We have a little bit of time still left.
Yeah. Can you move to the slide of result sections?
You mean this slide?
Yeah, yeah. So how do you differentiate between additive effects and the two synergetic effects in your results?
So basically, there is something called I think I forgot the name, but it was efflux pumps. So these actually exist in the bacteria itself, and they would basically inhibit the antibiotic. And this is something that bacteria has developed over time. However, I could not, due to the lab, I could not actually say for sure that these inhibited it. I could say that garlic and streptomycin together could inhibit more than just with garlic by itself. And that's how I figured out it has a potential synergy by comparing the combination treatments with the plant extracts by itself.
Okay. So this is you're done to the practical only. You got the values of practical, if I'm
Yes.
not wrong.
Yeah, this was all done practically, and I just looked at the data side by side. There is a way to quantify the synergy, but based on what I've done, it wouldn't have been possible to.
Did you compare your data with other review articles or research papers?
No, I did not. I kind of looked online and nobody had done this. I couldn't compare it side by side.
Why did you take the specific bacteria called Streptococcus? Why did you take any other one? Is there any specific one?
So Staphylococcus formed biofilms, which kind of make it harder to overcome the antibiotic resistance. Also, this aligned with the ISEF rules because I could only test about three bacteria, which was E. coli, Staphylococcus epidermidis, and Bacillus cereus. And I kind of wanted to take a unique approach and instead do Staphylococcus epidermidis. But Bacillus cereus, the problem with that one is that it didn't grow fast enough. I would have to wait even longer for it to grow to actually see the results. So I kind of went with Staphylococcus epidermidis.
Okay, okay, fine, sir. Thank you.
Awesome. Thank you, judges, for those questions, and thank you, Hashidi, for your answers. We are going to move on to our next participant. So, Hashidi, thank you so much. We'll stop your share, and we will have written feedback uh, in the future, right?
All right.
And then what we'll do is um, for the results for tabulation, we'll have those today. So we'll have those, by 1 p.m. today. So you'll hear about that. Awesome.
Okay. Thank you
Thank you
so
so
much.
much. Yeah. Bye, Hashini.
Have a
Great
great
work.
day.
You too. All right. And so now we have oh, I didn't realize that we had both you. I'll update the schedule, and I'll update the rubrics, but we have our next participants. Can both of you put both your names into the chat so the judges can see? And if you could also share your slideshow. Thank you. So we have Asmika and Evanthika who are going to be presenting, and I'll update the names of this. Uh, these two young scientists are going to be presenting a joint project. And so the way that the judging will work on this is that we're judging kind of a collective project. We're not judging them individually. Give me one moment to change that naming. There we go. All right. And same thing. So if you could update into the Excel files, judges, that would be lovely. And we will so Avantika and Athmika, we will have 10 minutes for presentation and then about 5 minutes for questioning. So we'll keep it at 15 minutes so we can standardize each participant having the same amount of time.
Okay.
All right. So yeah, give it a start. Thank you for being here and competing.
No problem. Good morning, we are Atmika,
and Avantika Tanike.
and today we will be presenting our project, HemaView, a novel way to detect microplastics in simulated blood. Microplastics have become an emerging concern in both environmental and biomedical research due to their increasing presence in human tissues and fluids. Recent studies have confirmed that microplastics can be detected in human blood, raising concerns regarding their potential health effects, such as oxidative stress, hormonal dysregulation, inflammation, and cellular damage.
Despite their growing concerns, current detection methods such as FITR and Raman spectroscopy remain impractical for widespread screening. These methods are expensive and require highly specialized laboratory equipment and often take several hours to days to produce results. Because of these limitations, our goal was to develop a rapid, low-cost, scalable, and clinically accessible method for microplastic detection in human blood.
Our research question was, how can a low-cost, standardized, rapid, and sensitive method for microplastic detection in human blood be developed to overcome limitations in current testing approaches?
Our hypothesis was that a non-red-based fluorescent paper strip test would allow for reliable microplastic detection in blood samples. Because non-red binds to hydrophobic plastic polymers, we predicted that the stained microplastics would glow under a blue excitation light and could be easily seen and quantified using basic microscopy.
Our methodology demonstrates the fluorescent properties of non-red in microplastic detection. Mays et al. Showed that non-red selectively stains hydrophobic plastic polymers. Additionally, Ernie Casola demonstrated that fluorescence-based detection is significantly faster and more effective than traditional spectroscopy methods.
These findings suggested that the fluorescent imaging could serve as a practical alternative to spectroscopic analysis while still maintaining strong sensitivity. Our project builds upon this concept by integrating fluorescent detection with automated image analysis into portable diagnostic platforms.
To build HemoView, we had three primary components: a digital microscope, a 495 nanometer ultraviolet blue excitation light to induce fluorescence, and a Raspberry Pi 5, which served as our computational processing unit.
The Raspberry Pi 5 interfaced with both the microscope and the image analysis software to automate microplastic quantification. This system was significantly engineered to maximize affordability and portability while preserving detection accuracy.
Our methods. We prepare non-red test strips by dissolving 2.5 milligrams of non-red in 250 milliliters of acetone solution. The chromatography paper were dissolved into the solution, dried in darkness to prevent photodegradation, and cut into standardized test strips. We tested three main plastics: polyethylene, polypropylene, and polyethylene terephthalate. We shredded them into three precise masses: 0.25 grams, 0.50 grams, and 0.75 grams. We prepared 27 containers of simulated blood and mixed each of these masses into the simulated blood. We took 5 0.25 milliliter samples from each of these containers, resulting in 135 total test strips for analysis. The samples were then applied to the non-red test strips and exposed to the blue excitation light. The fluorescence microplastic particles were then imaged using a digital microscope for further analysis.
To automate analysis, we developed Ponia. Ponia stands for Python-based OpenCV22 NumPy Image Analyzer. This Ponia processed each fluorescent image and quantified the number of microplastics present. This was needed for the subjective manual counting and enabled rapid sensitive analysis. Our results showed that our precision score was 96.7%. This means 96.7% of the detected particles were true microplastics. Recall score was 96.4%. This shows that 96.4% of all particles present were detected. F1 score was 96.5%, indicating a strong overall reliable detection.
We then validated HemoView against manual handheld lens counting. Across polymer types, HemoView consistently detected more particles than manual observation. One limitation of our study was that the image analysis was restricted to the microscope's central optical field. This is why analysis was limited.
On average, HemoView detected 78% more microplastics than manual counting, with a mean bias of 22.1 particles. Paired t-tests showed p-values below 0.0001, confirming superior sensitivity across all polymers. This shows that HemoView never underperforms. HemoView successfully demonstrates that low-cost fluorescence-based microplastic detection in blood is both feasible and highly effective. At approximately $2 per sample, HemoView offers a substantially more accessible alternative to conventional laboratory spectroscopy. Moving forward, our research will focus on determining the quantitative safety threshold for microplastics concentration associated with adverse biological effects in humans, because there is currently no universally established threshold level. To minimize positional bias, we standardize imaging viewpoints across all samples. One of our future goals are to get a higher resolution imaging system that enables full strip scanning. Thank you.
Awesome. Thank you so much, the two of you. So we definitely have time for questions. So judges, what questions do you have for Athmika and Avantika?
Hi, what microplastics did you use?
So we shredded all of like like the household items like yogurt containers and food containers, and we made sure that they were either polyethylene, polypropylene, or polyethylene terephthalate because those three plastics are most commonly found in our like blood and ovarian follicular fluid.
Got it. And then I'm assuming there's, a shredded mix of these plastics. How did you ensure that each one of your samples received the same mix?
We measured it on a small scale to make sure that each like it was close to 0.25 grams and measured it into all of the containers of simulated blood.
Got it. Is there any part of your methodology that ensured that, let's say you shredded a plastic bottle and I don't know what else is plastic, but say a plastic bowl. Is there any part of your methodology where you ensured that each sample gets 2% of the bottle, 2% of the bowl, instead of a sample getting 50% of a bowl and 1% of a bottle?
So we only used like one item for each plastic kind like type. So for like polypropylene, we used one plastic item. For polyethylene, we used one plastic item. So we didn't really quite mix it. We just had separate plastics.
And then what is the real world impact of your research? How does
So we're,
what does this research do? Mhm.
so we're planning to use this in a clinical setting. Like when you get your blood test, we want like microplastics like in your blood, that level to be included in your reports. And we also want this to be easily and accessible so that you can do this anywhere that you have.
Because based on like recent studies, microplastics pose serious health risks to like the human health, and it could also trigger biological changes that we can maybe never reverse.
Yeah, it was a good explanation you have given to this microplastic.
Sorry, could you repeat the question?
Yeah, yeah. What is the smallest size of the microplastic your paper strip assay can relatively detect compared to Raman spectroscopy?
So our plastic particles were less than 5 millimeters in size. So the smallest that we could have like was barely visible to the human eye, but we use it under a microscope. And Raman spectroscopy, it usually takes a longer time to process the microplastic particles that are present. So our method is faster.
Do you know anything related to LOD, like some form of LOD?
Sorry, we can't quite hear you. What?
Do you know full form regarding to the LOD, like we have used in this concept, no?
Yeah, so
LOD.
yeah, we've seen that those are still those results are sent to the lab, so they still take quite a long time to get the results back. But our project gives immediate results in just like a few minutes.
Can you elaborate LOD full form? May I know the LOD full form?
What?
Can
We
you
didn't.
explain a little bit more the difference between the process for LOD and the process of your device?
So our device, you can do this anywhere. It's a portable and scalable device so that you can use it in like large screening events, and it's really fast. You can get results in less than a minute or like a two. And but like traditional methods like LOD, they require like large like laboratory equipment, and it takes a lot of time for the results to come back, and that's not really accessible for like large scale screening practices.
How do you account for different types of polymers? Does fluorescence intensity vary significantly between the plastic types?
Yes. So we have this chart, and the fluorescence colors would match whatever plastic that we put, so that you can detect what.
Yeah, so as you can see, if you can see in this slideshow and the images on the bottom, we had the software like quantify and identify the different plastics, and we group them as fibers, pellets, and spheres based on their morphology.
The different colors that you see here, the yellow, orange, and the blue, those are based on the morphology of the microplastics.
Okay, so Neil, red is known as a bind bind to the lipids as well as the plastic also. So how do you ensure that fluorescence you see in the blood sample is actually a microplastic and not a natural lipid droplet?
So we'll use pre-digestion like protocols such as hydrochloric acid to make sure that we wash out all of the natural lipids and that we can only stain the microplastics. So we're making the human blood simulate the simulated blood samples that we're using right now.
So what is the percentage of the pollution that may affect to the environment?
Could you repeat?
Yeah. We can't quite hear you.
So what is the percentage like pollution can be compared to this microplastics?
The pollution of the microplastics? Is
Yeah.
that
So basically
Yes.
you are doing the experiments on a microplastic. So it is as compared to the nature, it is causing a pollutions. Plastic is causing the pollution. So you are
yeah.
using microplastic. So what detection you are taking based on that?
So you just like microplastics, because of the pollution on microplastics, they pose serious health risks. So we're kind of the pollution is kind of masked into the microplastics.
And plus, the community doesn't really know about microplastics. They only know about plastics recycling and all that stuff, but they don't really know the like the health risks that microplastics pose. And microplastics have been found everywhere, like in every life. Like if you open a plastic water bottle, that's releasing microplastics into the air.
Okay, thank you.
Thank you.
All right. So a follow-up question that I think would be good would be for the experiment that you were doing for the concentrations for the microplastics that you were going through and measuring or I should say using in your experimental design, how do they compare to the ambient concentrations that exist?
So our concentrations were taken from different studies done by scientists, and then we made the average like 0.25 grams, 0.50, and 0.75 grams to make sure that we mimicked what actually is happening in human blood.
Perfect. All right. Well, yeah, thank you. Super
Thank you.
direct answer. Thanks. All right. So we are at time for questions, and so we are going to be moving on to our next participant. Thank you so much,
Thank you.
Advika and Eventhika. Excuse me, I'm so sorry. Uh, we are going to be giving you the direct feedback from the judges. That will be for tomorrow, right? But we're going to be having the tabulated results by 1 today. So you'll hear back today about the overall results, but you'll get the more direct feedback a little bit later. But you're probably well practiced in all of this now at this point. Uh, all right, so thank you so much. Really appreciate you being here.
Thank you so much. Have a great day. You too.
Yeah, bye, you too. We have a new judge who is here. We have, oh my gosh, William, do you go by Bill or Will? I forget.
I go by Kyle. I don't know
Kyle,
why it
right?
says William. William. No
Okay,
one knows my first name. I publish as Kyle
I
Hamilton. I've gone as Kyle Hamilton. I have
I
KyleHamilton.com.
I know I got confused because it said William, so I was like, oh gosh,
Me
have I
too. I
been
looked down
calling?
there. I looked down there. I was like, oh, oops.
Yeah, I got nervous. I was like, have I been calling this person the wrong name for the
Oh my
entirety of
goodness.
knowing them? Okay, and the answer is no. So that's good. All right. Well, uh, so we had Markle and I lost the screen. So I think did Markle already bounce out? Yeah. So Markle, thank you so much for judging. We are your time is done if you need to go. If you want to keep judging, feel free to stay. I'd love that. Same for Likit. If you want to stay too, you're welcome to and keep judging. But I know it's very late for you, so if you want to head to bed, I understand as well. Um, so have a good day. If not, please stay. Whichever you decide. All
For
right.
the
Yeah,
for the
thank
rubric, we
you.
just fill that out and leave that in the shared file, correct?
Yeah. You just like
Yeah.
open it, add your 2 cents, and put that in there. And then Kyle, I will give you that folder. Oops, there we go. All right. So Kyle, you have the folder. And then yeah, so there you go.
Okay, thank you.
All right. Thank you. I really appreciate it.
Is
Okay.
there a file I need to look at or something?
Yeah, so you will see so
Oh, student
our
name.
next yes. So well and they're already made for you. So if you go to the
Oh,
schedule,
got it.
our next participant who is already here, so we have uh our next participant, and can you remind me how to say your name? Because I am learning how to say all everyone's name.
Yeah, that's perfectly fine. My name is Akshit.
Akshay. Okay, right. Okay. Miss Nath was helping me to try to say everyone's name, but uh I don't want to get it wrong. So we have Akshit who is here, and so he's going to be presenting as our next person. So we'll start very shortly. Appreciate your patience. And so Kyle, you will be judge one. So Akshit has 2 files, one that says judge one at the end and one that says judge two at the end.
Got it.
Right. All right. And then uh and then if you're going to stay Likit, then it would be great if you'd stay judge two and help us with Akshit.
Yeah, sure, sure.
Oh, thank you. Thank you. I really
Yeah.
appreciate it. Thank you. Perfect. Okay. So, actually, can you share your PowerPoint for us and then we'll get started. So you'll have 10 minutes. Oh, let me get my timer. So you'll have 10 minutes for your overall presentation for the content and then 5 minutes for your questions.
Perfect. I share my screen. Can you see my screen now?
Yeah, I can see my face on there and it's covering up some of your screen. So can you try a reshare and try just the instead of the whole computer screen, maybe just the one file.
Does that work?
So try stop share where it says that. Yeah, perfect. And then if you go to share screen again, and then try just like if it's open in a window, just do that one window, not the whole computer screen.
Got it. Okay.
Yeah. Now this is a skill for, corporate, international, US, everywhere. So you are developing a skill right now in real time.
Yeah.
Congratulations.
Thank you very much. Oh, how do I store that?
Perfect.
Right. Okay, that should be there. Okay, I'll just print a slideshow. And is your face
You,
gone or is it still there?
yeah, my face is gone. Before you can go into slideshow, you might have to say I'm not interested. I know my computer does stuff like this.
Okay.
Yeah.
Oh yeah, let me just put a slideshow.
All right. So whenever you want to start, we're ready to go.
Perfect. Is this fine if I just present like this? Or should I go with like, this is fine, right?
Yeah, this works.
Perfect. Okay. So, hi everyone. My name is Akshay Chipuri. I'm a sophomore high school student at University High School. I have done this research program under University of Arizona Star Lab program, and the research that I've done is the effect of feature caching on latency and accuracy in machine learning based credit card fraud detection. So, first, the research question that I have. Does using a feature cache with pre-aggregated transaction summaries reduce the interference in simulated credit card fraud detection system without decreasing classification accuracy? Okay. So my methodology that I used was analyzing existing fraud detection pipelines to identify latency bottlenecks in database access and feature extraction. And I also generated real-time aggregated transaction features on demand while measuring computation latency. I also trained and fine-tuned random forest or XGBoost fraud detection models using Python-based Python-based machine learning frameworks. I also assessed model performance and speed using accuracy metrics such as precision, recall, F1, and AUC ROC, and inference latency to compare optimization strategies. Okay. So, just giving a brief introduction is that credit card fraud is on the rise, especially with digital transactions, and there's a need for faster and more accurate detection systems. Although AI is widely being used for credit card fraud detection, system delays may result in several fraudulent transactions occurring before even action is taken. According to King Saud University College of Computer and Information Sciences, fraud detection is challenged by real problems such as class imbalance, data drift, the lack of real data, and high time complexity. Also, machine learning models are required to process transactions at a high speeds, but real-time processing and validation can cause delays due to continuous data processing and system integration. And if we see here from the US Federal Trade Commission at 2024, the number one top frauds were imposters, online shopping, and negative reviews, and um, businesses and job opportunities. 2.6 million fraud reports were issued and $2.5 billion were reported lost. That means more than 1 in 3 person were reported a scam who lost their money. And just for my hypothesis is if the transactions are processed through the cache feature pipeline, which is condition B, then it will show significantly lower mean latency, which is measured in milliseconds, compared to the on-demand calculation pipeline condition A, with no statistically significant difference in classification accuracy. So my independent variable is type of retrieval model, which is condition B and condition A. dependent variable is latency, which is time from transaction input to fraud detection output, which is measured in milliseconds, and accuracy, which is classification performance on a holdout test set. These are the four metrics that I've used was AUC, ROC, precision, recall, and F1 score. Constants, same ML model architecture that I use. Examples were random forest or XGBoost, and also same training data and same hardware/compute environment and same test transactions. Control group was my condition A, which is the traditional database query for each transaction simulated real-time feature calculation. Experimental group, which is the condition B, this is pre-cached, pre-aggregated features stored in memory. So something about my information, my data variables. I have used a Kaggle data set, which was um, uh, so the Kaggle data set was MLG. It was MLG CBU, and it was 2013 data set. So that data set did not contain any missing data from any 31 columns. So the 31 columns were time, V1 to V28, and amount and class. V1 to V28 are components obtained from PCA, which is Principal Component Analysis. The original features are not provided due to confidentiality issues, but PCA transforms the original features into a new randomized set of variables. Time. So this is the number of seconds elapsed between each transaction and the way first transaction in the data set. Amount. This feature can be used for cost-sensitive learning. And class. Class means a value of 1 indicates fraud, while 0 indicates legitimate. Ratio of legitimate to fraud. So basically, in the transactions, there was almost 278,000 transaction 200,000 in the data set. There was 278,000 transactions, and every 570 legitimate transactions, there was one fraud. And here we can see in the data set, there was almost 284,315 legitimate, while there's only 492 fraud in that entire. And we can see here the time distribution for transactions and fraud. Here we can see from all the hours from 0 to 45 for almost approximately 50 hours, which is we can see the legitimate around 22 hour to 33 hour. We can see there's a dip in the legitimate transactions. So maybe during that time, there was more fraudulent transactions happening. And we can see during 35 to 50, there was pretty constant 8,000 transactions being legitimate. And our peak was 22 hours, which was almost nearing 9,000 legitimate transactions. And here's if we see for the fraud, at the 12th hour, there was almost 40 transactions flagged as fraudulent. Of course, we do see there's not that many fraudulent transactions. However, even one delay, even one transaction that can be like exposed as fraudulent, that could still damage any institutional bank's reputation, and any commoner's accounts. So every single transaction is important to be viewed as positive, not negative. And here we can see, so the three models that I've used was logistic regression, decision tree, and random forest. And the confusion matrices, which means how the model was confused depending on legitimate versus fraud. If we see logistic regression had around 56,851, decision tree had 56,857, random forest had 56,859. All of those three were very similar. They recognized the legitimate transactions pretty easily. But however, for logistic regression, it recognized 63 fraudulent transactions, while decision tree did 75 and random forest did 79. So logistic regression is a better model compared to decision tree and random forest for this data set, because it was able to actually recognize the fraudulent transactions as fraud, other than decision tree and random forest recognizing fraud maybe as transactions. So maybe as legitimate. So that was the confusion matrices between these three. And yeah, so these are my four key performance indicators that I've used was F1 score, recall, precision, and AUC ROC. Here I did the same thing again for the models, logistic regression, having almost a 0.7241 score for F1 score, and decision tree at 0.83, and random forest at 0.86, which is the highest in that category. Random forest really did good on F1 score. And also for the recall score, we can see random forest at 0.8. For precision score, we can see decision tree and random forest were pretty similar, almost close, but random forest did have the upper hand at 0.94. But for AUC, ROC, logistic regression has 0.95, which ultimately beat decision tree, but it kind of failed to random forest. The problem is with random forest is it takes a lot more time to process the transactions. That's why it's more consistent with results. And logistic regression is the more faster approach, but it's more quantity based, but not quality based, while random forest is quality based and not quantity based. Decision tree is in the middle of these two ideas, and that's why I was able to give a very good score compared to both. And so these are my V1 to V28 components absolute difference. So we can see here V3, the difference basically means how many how many fraudulent transactions were registered in these. So we can see V3 has 7 transactions and V14 also has 17 transactions. And we can see it goes in a downhill. So this is good. This is very good in the data set that the transactions have been flagged in a downward because we do want to see less and less fraudulent transactions every here and there. So it does prove like um, our systems, any data set that was this data set that provided, which was a randomized data set and was a prosthetic one. It was not a real one. It's a made up one because due to confidentiality issues, we cannot possess real data set without having the consent. So that's why this is a synthetic data set. And we can see here from all V3 to V28, it does like after V16, it does show a downward trend. So the versions 21 to all the way to 28, they have like improved, I guess. They have improved and saying that they were able to like not let they were able to identify fraudulent transactions easily. And here, in the training and prediction time for these 3 models. So the training time for random forest took almost 260 seconds. This is what I said by the latency thing. Random forest takes a lot more time compared to the other two. Logistic regression only took 3 seconds, and decision tree took 20 seconds, while random forest took almost 3 minutes and 20 seconds. And for precision time, same thing. Random forest took almost a second, while logistic regression took almost less than even a second, 0.04 seconds. And decision tree, I think, even scored even less, 0.02. So we can see here for random forest, it takes more time and predicts more and takes more time to predict. So that's why although it is being consistent, it does have that latency issue. And in that time of period, anything like any system can have a delay, any malfunctions that could cause even more. Thank you very much. That could happen anything. So that's why this random forest is a bit more taking more time. And the caching speed up, which is so the baseline no cache took an average of 70 seconds latency issues. And with the binning, so this is a feature binning. So using one feature in 5 bins took less than even a 0 second. I mean, yeah, it took less than even 0 seconds. And then same thing for feature binning, 2 features 5 bin, 3 features 5 bin, and 4 features 5 bin. They all scored very good. And the speed up over baseline, this is how much it was able to speed up compared to the baseline. Feature binning, one feature in 5 bits had it almost 800 times the speed up over baseline cache. And I'll just move on to the results and conclusion. And so the best viable strategy that I found was to use similarity cache with precision 0 configuration. Its average latency was 57.018 milliseconds, and having a 1.0 times faster speed up than the regular method. Another strategy is 4 features 5 bits, having 0.1332 seconds, milliseconds average latency, and 436 times faster speed up, but scoring a 0.72 F1 score. 4 feature 5 bins results in 625 different combinations and has a decent F1 score. With more features and bins, the possibility of an increase in F1 score and a decrease in average latency. And these are my references that I've used throughout my research. And yeah, that's pretty much it.
All right. Thank you so much, Akshit. Judges, we have about 3 minutes for questions. So let's see if you have one question each or something like that.
Yeah, so I guess I have a couple quick questions. So when you were looking at the fraud, did the data set allow you to know how much money each transaction had? So for example, a model might have a better performance rating, right? It might be able to detect fraud more often, but if it's detecting for example, if you had like 50 transactions that you catch that are $100 each, they're all small, you're only saving about $5,000 to the company. But if it was a really accurate model that's catching higher levels of fraud, so like, I don't know, a dozen at like $9,000 each or something, that's like over 100 grand, right? So if that data is available in your modular data set, that'd be really interesting to look at, right?
Yeah, that's actually one of my ways to improve this research project and continue my research. I'm trying to contact my professor for more continued research, and we're trying to bring more a stronger model and maybe even a more available with more data sets that has more transactions that we can probably use. So that's actually one thing I do want to pursue. Yeah, thank you very much for
Yep.
that suggestion.
Another quick one might be also looking at median versus mean time, because a
Mhm.
lot of this data set isn't going to be normally distributed, so it's spread out, just like how
Yeah.
your chart here shows. So it
Mhm.
might be kind of interesting to see what the most average kind of time is going to actually look like, and seeing if there's like large outliers. So for example, if there's how do you think a difference in timing would affect a model's output performance? So, for example, if there's delays that are not related to the data set. So, if you have like a transmission delay because it has to go through a satellite, for example, or if it has to go through a different method or a different system to get routed to the actual credit card processor, do you think that kind of delay would have an effect on the overall model performance, or do you think it wouldn't be baked in at all?
I could definitely see the time communication, the time latency could happen throughout this long process. And while the main issue that I do want to encounter in this project was to like accurately like um reduce the latency issue at least by any milliseconds, that would really help. So I do want to like reduce anywhere that I can reduce any time constraints. So yeah, the complexity process. The thing is for this research program, when I originally did the research question, it does restrict me from going even complex process. That's why I have to be a little more careful and like doing a little simpler and constructive process so that I could prove that how should I say that there is a possibility to speed up. However, with more investing in technology and AI and all that, there is a definitely a big chance in the future that with this big of a complex process, there will be a decrease in average latency time.
Very good. Thank you.
All right. One more question if we have one.
Yeah. How did you design an adaptive catching strategy that balance the latency and accuracy while in training?
I'm so sorry, I couldn't hear you.
Yeah, can you talk a little closer to your mic?
Yeah, how would you design an adaptive catch strategy that balance latency and accuracy dynamically?
You're saying how would I like establish a caching strategy that would balance both fraud and legitimate?
Yeah, yeah.
Okay. So what I did was I used these binning strategies, which is one feature, five bins, and two feature, five bins, three feature, five bins, four feature, five bins. The more features and bins you have, it takes more like one feature, five bins is only like five combinations, but two feature, five bins is 25 combinations. Five bins and three features is 125 combinations. Five features and four features is 625 combinations. The more features and bins you have, the more combinations you can have, and that results in more time, like more time will be taken to process, but it will show a better speed of process and better accuracy results. However, my IDE, so my integrated development environment, could not support more than like 625 combinations, which made me to restrict at four features, five bits. But with a much more better model, better IDE model, and a better data set and a larger transactions, I could definitely go on to make a 10 feature fit of 20 bins, which could result in almost 100,000 different combinations that would really help how we can analyze how to reduce latency, and we can see which combination or which area is the latency being marked.
Yeah, okay then. Thank you.
Yeah, thank you very much.
Awesome. Well, thank you so much. We really appreciate you coming and presenting and being a participant for the Research Innovation Expo, Akshit. So you will get the feedback sometime after the event, and then the results will be tabulated today and sent out. So thank you. And
Perfect.
then we have our next participant who was going to be sharing their slideshow. So we have Ria, who is going to be our next presentation.
Yeah, it's really nice to meet you all. So my project was SMS scam protection using feature engineering across ML architectures using real-world user validation. So imagine the feelings that run through a person's mind when they receive a suspicious scam message: panic, confusion, fear, lack of trust. Now imagine how those fears multiply when I tell you that in 2024, Americans reported losing over $470 million to smishing. Specifically, smishing disproportionately impacts seniors, veterans, and youth. But existing models are trained on scarce, imbalanced data sets, producing false predictions with very little explanation. This heavily limits user and trust and adoption of scam detection systems. So my problem statement was that, can we improve SMS scam detection accuracy and explainability for elder populations and enable adoption using feature engineering and SHAP analysis across multiple machine learning models? So to do this, I explored multimodal testing using SHAP analysis for detection, semantic feature engineering for augmented training, developing a full stack and well-deployed robust scam detection system that promotes explainability and accessibility, that I couple with a community forum that serves as a repository of reported scam messages, and then also evaluate my solution with user validation focused on the senior age demographic. And my hypothesis was that in doing so, a multimodal detection system with SHAP analysis and feature engineering would outperform existing commercial spam detectors in accuracy, recall, and cost efficiency, driving higher trust and adoption in older populations. So to do this, I explored three different ML models that were each chosen for their efficiency and ability to extract contextual understanding, and also from papers that I had read in the past that had explored different scam detection models. So the first one I used was logistic regression model, then an XGBoost model, and an LSTM model. Each of these models was trained on a dataset with 10,000 SMS messages with balanced classes of ham, spam, and smishing classes, ham being safe messages. And then each model was evaluated using accuracy, precision, recall, F1 score, edge case testing, and confidence analysis to best judge my models and see how they can be improved. Of course, I won't be going into all of these metrics today, just the most meaningful ones to show the difference between their performance. And then I used SHAP to extract Shapley values that would determine feature importance of each token in a text towards the final prediction. So to go over the model pipeline a little bit, I would use SHAP to extract words from each of my messages that correspond to smishing classes, spam classes, and ham classes to then determine the density in each inputted text of how many words are ham, spam, or smishing as a feature. This is something that hasn't been done in past papers that I really wanted to introduce here. And then I also coupled that with other features such as punctuation, unknown characters, capital letters, repeated words, and other semantic features like that. And then for some models, I also use the TF-IDF vectorizer to extract quantitative and frequency features to improve detection. And then all of those features were fed into the model for training and prediction. And then once the detection was gained, as you can see in my figure 2, I would use SHAP values to extract the feature importance of each word and then also give the characterization that those results I would all store in a scam detection dictionary, which would store the message, prediction, and meaningful word features. Then once those results were extracted from my model prediction, all of my code was integrated into a web deployed scam detection system that used HTML, CSS, and JavaScript as my tech stack. And then I used Flask to handle routing between pages and send requests to my Python models and pipelines. And then that meant that a user could upload a message to my system and then get a prediction as smishing, spam, or ham And then they wouldn't just get the prediction from our model, but they'd also receive the confidence of the model, suspicious words found by SHAP, and also high confidence categorization of the message type, as detected by a second logistic regression model that I had trained using categorically labeled spam and scam messages. So this system also allows our user to pick which model they wish to use for their detection. As I listed above, I provided 3 models, and that creates a multimodal system for the user. And then finally, I added a publicly accessible community forum where you can report messages that you found as scam or spam to a forum where that not only raises awareness of spam and scam messages, but also contributes to a growing repository of messages used to augment training for my models, but also future models. Then to evaluate my system, I compared my system against 4 existing commercial scam message detectors: F-Secure, Nickel, Ask Silver, and Texter. These can be found online with the same 35 messages that were labeled from my dataset. I evaluated them all using accuracy, precision, and recall, and then compared them against each other. And then to evaluate the effectiveness per my users, I surveyed 35 anonymous individuals of different age demographics on their experience on a 5-point Likert scale across 4 different key areas: navigability, clarity, understanding, and trust. So then getting into results, we found that the logistic regression model, once it was augmented with feature engineering and the text TF-IDF vectorizer, significantly outperformed all other existing models, even though they were deep learning, which is something that builds upon past work. And then you can also see how feature engineering improved that detection on logistic regression. As you can see that there were some messages that were classified as false positives or false negatives in these lighter areas in figure 8a that then were predicted correctly as the darker areas increase in value in the second figure 8b. Then, going into our evaluation of commercial models, we found that compared to the 4 commercial models, our system significantly outperformed them in accuracy and recall, with an accuracy of 82% and only a 6.8% error. The proposed system also was tested using the multinomial test, revealing a 0.011 p-value, meaning that the proposed system performs significantly better than random prediction, and it truly does outperform existing commercial models. Then, from our survey of 35 individuals, we found that the meeting score for trust and clarity and referral were all 4 out of 5, and then 5 out of 5 for understanding and explainability. We chose a median as our a measure of center because the mean was skewed by a minimum of 2 in the survey data, which only appeared once the data was checked. So that meant the mean was skewed, and we used the median to relay our results. But this shows a very good acceptance of our system across multiple different factors and just demonstrates the effectiveness of our system. So then discussing our results a little bit, we found that logistic regression had the highest performance out of all tested models and commercial models with an accuracy of 98.2%, and feature engineering significantly improved that classification across all classes as seen in our confusion matrices, advancing classification capabilities. And then with our survey and commercial comparison results, we found that survey responses indicate a strong user trust within all of our key areas. And 80% of our survey responses came from a 45 plus age population, indicating a benefit to an at-risk demographic. We also found a positive significant correlation between explainability and trust, which shows that when a user is given an explanation of their result, their trust in the system increases significantly. Our proposed system also outperforms existing commercial models in accuracy and recall, demonstrating a very strong detection capability of our solution. So to conclude, our model significantly outperforms existing research and commercial detectors, offering a very valuable technique in advancing smishing protection. And our proposed system was very successful in practice, receiving a 4 on trust, clarity, and referral, and a 45 plus age demographic being the most to refer our application. Additionally, our strong positive correlation, which is something that our app very heavily promotes given its explanations, shows that there should be a further resolve to strengthen explainability of scam detection systems and improve that correlation to increase adoption. And overall, the research indicates the potential for a full stack scam detection system that's backed by machine learning models and augmented with feature engineering in protecting vulnerable populations from smishing and providing a trustworthy, intuitive tool. And then for future work, what we plan to explore is OCR implementation, which is where a user can upload a screenshot of their message to be detected in the same way, collecting more survey responses to further the like understanding of user acceptance of our model. We would want to explore mobile application and extension onto existing SMS services to increase the accessibility of our system, increasing overall accuracy by using the data collected in our community forum, and then also driving awareness in senior citizen communities, and then also other vulnerable populations like veterans and youth. And then these are the references that I used.
All right, perfect. Thank you so much for telling us more and about your research. We have about 4 minutes for questions from the judges. So judges, take it away. I think we have one from Gosia.
Just show myself. Thank you very much for your present. Can you hear me?
Yes.
Cool. Thank you very much for your presentation. Very interesting. One thing caught my attention when you were saying, I think around slide 10, you were saying that explanation increases the trust.
Mm-hmm.
And I'm very much interested here to learn from you whether you took it into consideration or you consider to take it consideration. Does it matter if this explanation is true? Or maybe if somebody speaks with very confidence and talks, an explanation doesn't make much sense, but if somebody talks with much confidence,
Right. So
can they still convince person? And did you take it into consideration? Does it make sense to you? What I'm
yeah, so yeah, and it's definitely dangerous if a wrong explanation is given and then it convinces the user of the opposite. However, for our explanation trained model, we did evaluate that on accuracy, precision, and recall, and those received high accuracies in the 90s, which means that there is very low risk of like false explanations.
okay? Thank you very much for that.
Hi, so when you were looking at making your models, were there any variables that you thought would have been useful in when you actually were creating your modeling? Were you thinking of anything that going back you may have wanted to add, or any possible interaction effects that would have been interesting that you may have thought of?
Do you mean in the training or in
Yeah,
the final?
in the training.
Yeah, I think in the future, I obviously want to explore even more features. I explored like pretty standard semantic features such as punctuation and unknown symbols and features like that that are very standard in semantic language. And then I also explored a very innovative one using SHAP for more contextual features about the class density. But I think it can obviously be improved adding even more features. To evaluate which features actually were meaningful, I used the mutual information score. So for example, I did explore potential like words that were tied to like finance or authority type of words. However, when I calculated the mutual information scores, those features were actually not meaningful in the prediction. So I think that's also something to consider when deciding whether to add more features and to improve and augment training, whether they're actually meaningful or not. But obviously, I'd want to explore even more in the future to augment my training.
And if you could add one variable, just if you could just add one to your model that wasn't in your data set, what would you add?
Um, I added the one about density of classes. One that I could potentially add is their like word density that tie into like the classes from my categorizer model. So like how many words are like from each of those classes that my other model uses, that's another feature that I could potentially add. However, I'd want to judge how meaningful that is, again, using the mutual information score.
Very good. Thank you.
All right. Well, we are at time. So well, I mean, technically we have 30 seconds. So does anyone have a 30-second question or that you want to ask?
What part of this was the most fun?
For me, I think actually collecting the survey responses was the most fun because I actually got people using my app and seeing what they said. And I also had a question that was like additional feedback or any words about my app. And I got really positive feedback about this really makes a difference. And I even like surveyed some other like people in the older demographic anonymously, and they like came back with like this really was meaningful for me, so it really meant a lot.
Very nice. Thank you.
Perfect. All right. Thank you for judges, and thank you for Ria for your presentation. So Ria, you will get feedback, uh, direct feedback from the judges and after the event, and then we will have the tabulated results coming out today, ideally by 1. And then we have, so Ria, have a great day. And then judges, we have one more presentation that should be.
I just have one question.
Oh, yeah, sorry. Yes.
So the awards ceremony is at 1, so we'll join this again.
Uh, yeah, we can join this. That's a really good question. I don't know if we fully like anticipated what we were doing, but yeah, that's a good idea. So I'll send out a message to the virtual participants and have everybody join this again.
Okay, thank you so much for your time.
Yeah, thank you. Yeah, we're running this the first time virtually as well, so
All good.
Yeah.
all right. All right. So we are tabulating a little bit. So we have one more presenter. Um, and then the two of you able to input your information into the Excel file?
I
Yeah, I'm
wrote it
almost
down.
finished. I'm just putting in the comments right now.
Okay, perfect. Thank
Oh
you.
yeah, I
Really
just put the
appreciate
quote.
that.
I didn't put the comments.
Okay.
Um,
Okay. Glosia, thank you so much. My gosh.
Not a problem. You can keep me in mind for if it's weekend. You usually can keep me in mind as a yeah extra person. Like probably it's better a little bit better tell me before.
I know.
Yeah.
Well, yeah, I mean,
Yeah,
I
Sarah,
publicized the event and everything, but we definitely were, well, we had everybody, but then one of our judges who was during this time slot literally canceled this morning and asked to move to another one. So I was like, crikey. So
I think somebody, I don't remember one. I think Christine was doing like a reserve or something, just in case.
it's okay, Sarah. You saved me from a quilt auction today. So my wife is out
Oh,
there, like I gave her money and she's like, "What do you think of this quilt?" I'm like, "I can't right now. Just pick one."
sorry, wife, cannot do this. All right. All right. Quilting. That's great. Okay.
Yeah, it's a fundraiser. So it's a bunch of older women paying hundreds of dollars for these handmade quilts. And I'm like, okay,
Oh,
well,
but I mean, that is shit.
it's yeah.
That's
She
legit.
sees one now that has all these red-winged blackbirds on them, like they were on campus, and she wants it. And I'm like, no, we can't spend $600 on anything, let alone a quilt.
All right. Um That's a lot. And then I and then Kyle, you're and Gosia, this is not a statement. Oh, good. Marcia is here. Okay, good. We have our last participant who's here. So judges, if you are able to finish up your comment that you're writing, we have our last participant. All right, so Marcia, whenever you're ready to start, and then Kyle, you'll be judge 1 in that folder. Okay. For Marcia Nath, or I think I put Marsh, but it's supposed to be Marcia. So apologies. Let me fix that. And then Gosia. Here is your file for her. All right, and then whenever you are ready to share your slideshow, or I think you prepared a poster, right? So
I did, yes.
yeah, so if you want to share your poster, and then you'll have 10 minutes for your presentation itself, and then you'll have 5 minutes for questions.
Okay. Let me share my screen.
Perfect. So just start whenever, and I'll start the timer whenever you start.
Okay. Sorry, the Zoom bar is covering the thing. Okay. All right. Good afternoon, everyone. Before I dive into my research presentation, I want you to consider the staggering statistics. Iron deficiency can affect nearly 40% of adolescents and young women. But here's the real kicker. In a recent 2024 study, over half of patients diagnosed with iron deficiency still hadn't resolved the issue 3 years later. We are looking at a public health crisis that is often invisible yet leads to chronic fatigue, mood changes, and developmental hurdles. I'm Marcia Nath, and today I'm sharing my research on how we can use a tiny transparent organism, the nematode C. elegans, to unlock better treatments for human iron regulation disorders. So, why did I use a worm? Well, C. elegans and humans share more genes and characteristics alike than you think. These worms share key iron regulatory mechanisms with us, including the use of ferritin protein for iron storage. In fact, they have 55 to 60% genetic identity with the human H ferritin. While previous studies established this link, there was a major research gap. We didn't fully understand the specific relationship between exact iron concentrations and the resulting behavior and physiological impacts. My goal was to fill that gap by testing how varying iron levels, specifically 33.6 micromolar, 66.6 micromolar, and 100 micromolar iron concentration affected reproduction and survival. To investigate this, I established 3 experimental groups with those concentrations I just mentioned, low, medium, and high iron, against a control group of standard NGM agar. I tracked 2 main metrics across 16 different plates, fertility and survivability. And with fertility, I wanted to ask how can 5 to 6 eggs hatch and successfully build a new generation? And for survivability, I want to test how well adult worms survive when placed directly into these iron-rich environments. I used ferric ammonium citrate for the iron source and maintained the worms on a diet of E. coli OP50. However, there were specific challenges and limitations to this. The high iron concentration actually turned the NGM agar, dark, making it really difficult to spot the eggs and worms under the microscope. In my results,
I predicted that my hypothesis was that both deficiency and overload would reduce success. In the control and low iron groups, the groups performed similarly, showing consistent egg production and rapid movement. In the control fertility test, I saw about 200 to 250 eggs by the end of the observation period. However, in moving into the medium concentration at 66.6 micromolar, things changed. There was slower movement, visible through sparse worm trails, and a significant reduction in reproduction. And at the high concentration, the impact was most severe. I saw the most negative impacts on both survival and reproductive success. Interestingly, one of the sample size of the high iron group showed higher reproduction, but I wasn't sure if it was due to fungal contamination, which might have skewed the results. However, what really fascinated me wasn't just like the hatch or die data, but the behavioral changes that the worms experienced. Typically, C. elegans clump together when they run out of food. In the control group, they displayed natural clumping behaviors when food ran low. However, the low iron group showed significantly less clumping. This suggests that iron levels don't just affect physical health, they may actually influence social and behavioral patterns. This kind of mirrors what we see in humans, where iron deficiency is linked in mood and cognitive changes long before it ever becomes anemia. For research applications that I want to further continue, my research confirms that iron regulation has a very narrow range of tolerance. To bridge the gap for those 58% of patients whose deficiency goes unresolved for years, I want to explore the development of a transdermal iron regulation patch. Similar to an iron patch, this could provide regulated, consistent iron delivery to the body, bypassing the absorption issues often found in oral supplements, which most people can or cannot take. Oral supplements typically can cause other side effects when the person's taking another medication, or they can just not be suitable to take in general. That's why most people have to resort to the option of IV iron. So with IV iron, that's the most effective treatment that they can get. However, it's really expensive, and most people that are affected with iron deficiency are low income and are in countries where the socioeconomic living status is developing still. Along with further research, I want to conduct qPCR testing to look at the molecular level of these changes to increase my sample sizes to further validate these findings. In conclusion, iron deficiency is a global problem, but by looking at it through the lens of a 1 millimeter worm, we might just find the high-tech solution we've been searching for. Thank you.
Sorry,
Yes.
I lost my mic for a second there. Really sorry about that. All right, so we have about 4 minutes for questions. All right, so judges, what questions do we have for Marcia Gosia?
What's the most interesting what you found out from this study?
So I mentioned before, a lot of studies didn't really investigate the behavioral changes in C. elegans that I've read. They were really looking at the molecular level and establishing that specific genes were homologous to the iron storage pathways in humans. So really looking at their behavior and qualitatively analyzing them through the microscope was really interesting to me. And seeing as how they were moving unusually in the iron, I want to explore like more on that and specifically have more sample sizes in order to investigate more and see how specifically iron affects movement of the C. elegans.
So if I could follow up, is it something that surprised you? Something that you didn't expect to see?
Yeah, I didn't really expect to see like the iron affecting movement. I was expecting them to like move normally within the iron, but I really wasn't considering any behavioral changes that would have happened with them in the iron.
Thank you.
If you were going to
Okay.
design an experiment for looking at how to at the behavioral changes, how would you design it in a way that you could expand this project?
So, one of the limitations of my project was the ferric ammonium citrate itself because it was so dark in color and I couldn't really see the worms and eggs themselves. So, to start off, I would probably get a better microscope or microscopy if I have access to that. And I would set up more sample sizes because I was only one person. I was looking at them individually and only looking at them in certain time spans. So I would establish more time spans like 24 hours, 48 hours, 30 minutes, or 1 hour in order to see those behavioral changes that the worms are experiencing because it's really just something I haven't seen before, especially with C. elegans because they're typically considered a model for genetics.
And how do you think that would impact the field? Like if you came out with a paper and you show that there's behavioral changes in the way they're interacting based on the density, do you think that would have any kind of impact on maybe the genetics or in how the research of these animals would go forward?
I think it would have a bigger impact because, well, this research was done a while ago in around early 2000s. So coming out with more C. elegans and iron research would specifically tailored to movement would add a new perspective to C. elegans and iron themselves. And I think for specific impacts within the field, more scientists would probably want to look at like the behavior of C. elegans themselves rather than just analyzing their genetics at a molecular level. There's been so much research into the genetics that looking at their movement wasn't really considered, and seeing previous research studies that didn't really account for that is just really interesting and something I was excited to see.
Very good. Thank you.
Thank you.
Perfect. All right. So I think we're at time to keep everybody consistent for all the participants. So, Marcia, we are, imminently tabulating for the award ceremony, and then we will get the direct feedback in terms of the rubrics out to the students after the event, so next week. And, thank you so much for presenting. We really appreciate it.
Thank you so much.
Yeah, thank you. Okay. So I am going to put this up. So to you wonderful, wonderful judges, if you have
this program or from myself. Now, I've probably seen patients and companies given me the confidence I
sorry there. If you have any specific things that you need to finish up on your, drafting for your things, wonderful, right? Please finish that. But otherwise, thank you so much for being here and helping us out with STEAM NHS on our Research and Innovation Expo. Thank you again. And if anyone wants to help stay and help me tabulate in another file, I will welcome that graciously. Um, otherwise, no worries. So Gosia, like I know you're in a different time zone. It's probably,
It's 14 in the evening.
yeah. So yeah. So have
Yeah,
a
pretty
good.
fast. I almost finished my comments are very brief.
That's okay. That's okay. I think that's fine. They're gonna get plenty for me. Don't worry. So
Oh, okay. I was like feeling really bad. There's like chunks missing out of mine where I'm like, oh, I
I
gotta
cannot
go.
that good because then have less. Frankly, if it's true, if you Kigakus and my head was not so much into that, yes, at the moment because I joined late and yeah. So
yeah,
it
no,
was difficult. It's difficult
you're
for me to write
perfect.
really a lot.
No, and mostly it's just, if you're able to give an evaluation in terms of
Yeah.
a number that's more helpful. And we have many judges who were helping us today from completely different backgrounds, so it's perfect. Thank you again.
Good. Thank you very
Yeah.
much. And yes, I see you again at some point.
Yeah, lovely. Thank you.
Not a problem. Thank you. Bye.
Yeah, bye. All right. Let's see how. So I figured out how to do a live stream for the first time ever. So that's cool. Um, she says loosely. Let's see. Live streaming. Yeah, it's wild. Okay. Yeah, I don't know, Kyle, if you want to help me add things in.
I can
You're
give you a
kind of
quick hand.
yeah, because you're kind of like me. You're like, data data nerd. Um,
So how do I, want me just to add up the two scores from each one of them into another spreadsheet or something?
yeah, here's that.
Got it. Okay. Yeah, I'll just add them up real quick for you.
Yeah, I haven't added anything for Marcia. So if you handle her, that would be like super helpful.
Got it. I'll take care of Marcia right now.
Thanks.
For sure.
Hello, my name is Yaratsi Lauren. I've learned a lot in my journey with this project. At the beginning, it was complete confusion. I had no idea what I was doing or what to expect from this program or from myself. I quickly realized that the project can stem from simple everyday objects such as stress-relieving toys. I've probably seen them around in offices and schools, but I realized as I gathered sources that there actually isn't a lot of research backing their credibility. I was surprised given how much we take for granted that the stress toys do work, and organizations and companies hand them out to help in the workforce. This showed me that I can't always take everything for granted and that you should always be questioning your surroundings to find more answers, even to things or ideas that you think are certain. This project has allowed me to expand on skills such as presenting, writing, and researching. It has given me the confidence I need to continue researching and innovating past my high school years and into college. Thank you.
Judges are so different.
Yeah, I'm looking at both of our scores. And I'm like, oh wow, we are scoring these people radically different.
That's why we do an average.
What happens when you make a psychologist grade a biology paper?
Totally, right? Right. Yeah, it's like we should do a power analysis of how many judges you actually need, you know? Um, could get a reduce your like 95% confidence interval. It's nice to talk stats to somebody who gets my jokes. Okay.
I haven't been able to have a good stats joke in a while. Like,
yeah,
it's been I don't know. I do workforce stuff now, so I'm like, all I talk about is apprenticeships. I'm like, man, I should have been a plumber.
Plumber.
Okay, so I got all the ones from this session entered for Access, Raya, and Martha.
Okay, excellent. Thank you. And then I'm just averaging. Did you do an average too?
Oh, I did not. Okay, let me do it real quick.
Yeah. Thanks. I appreciate that.
Yeah, for sure, for sure.
Not hard. I probably even have an average somewhere that you could steal it pretty easily. Clearly, I made this like really beautiful uh, Excel file that no one used except me. And then we need it for Athnica. Is that right? Can you double check that I entered this right? Can you
Which
double
one?
check the Athmica and Eventica? Is that right? Like, is this number right?
And
Did I
like,
bold it?
oh, sorry, I don't have you on which cell are you on? Oh, BK 24.
Yeah, like did I enter the
Yeah, that equals 69.
yeah, but I'm trying not to say the numbers out loud.
Oh, sorry.
No, but I mean, is this range, did I enter the
Yeah,
values
yeah.
correctly?
Oh, yeah, yeah. Let me just double check it real quick. Yep, everything looks correct.
Okay, that's so interesting. I just don't get it.
Oh, I see what you did. Okay, I should do that.
Did I do something?
No, no, no. It's a me thing. I was just like, oh, you merged the cells so they look pretty.
It's me being something.
I
Okay.
quickly realized that the project can stem from simple
All right, so we
everyday
got
objects
Marcia.
such as stress relieving
Perfect.
toys. You have probably seen them around in
Oh,
offices
let
and
me
schools,
average
but I
this one.
realized as I gathered sources that there actually isn't a lot of research backing their credibility. I was surprised given how much we take for granted that the stress toys do work, and organizations and companies hand them out to help in the workforce. This showed me that I can't always take everything for granted and that you should always be questioning your surroundings to find more answers, even to things or ideas that you think are certain.
Really?
This project has allowed me to expand on skills such as presenting, writing, and researching. It has given me the confidence I need to continue researching and innovating past my high school years and into college. Thank you.
Like that shouldn't be
Okay, I've also filled out the rest of the range with the averages for you.
Thank you.
Sure.
Well, some of them have 3s and some have 2s.
That's okay. I'll just do the 2s and I'll go through and do the 3s real quick.
Okay, yeah. Maybe we'll get this entered properly.
2. Yep, that one's correct. And okay, yep, they all look good. The only one that needs something is AU, columns AU through AX, just because there's nothing there. They say divide by 0, but once they get some numbers in there, it'll be fine.
Hey, you.
Oh, I see what happened. Let me go ahead and unmerge that. Go and then add the auto sum back. Never mind, I fixed it.
You're amazing. All right. Well,
And just double check each one. I like how like I know the computer did it right, but I'm like, man, they're kids. I'm not going to mess with this.
yeah. And I'm actually just gonna change this one because that actually gets merged, and this range goes to AX. Cool. Yeah.
Oh, they're
And
all together.
yeah, it's a group project, but they
Oh, okay.
theoretically,
Fair enough. That's all it needs to. Like, okay, cool.
yeah.
Anything else you need?
No, that's super lovely. Thank you. Really
For
appreciate that.
sure. If you ever need anything, always feel free to reach out.
Yeah, no, this was great. Really, like, also super fun. So we had like a bunch of UC Merced people, which was like honestly like crazy, like really, really crazy. Um, but yeah, well, thanks so much, Kyle. I really appreciate it.
For sure,
And
Sarah. Anytime. Thanks
yeah.
for saving me from a quilt fair.
Oh, well, fun times.
It's
All
not that
right.
bad. It's a quilt auction, but it's still it's my wife is the youngest person in that audience. So
Hey, we can't change what our soul is and what our hobbies are.
I know what
So
I married. Thanks a
yeah,
lot,
I know.
Sarah.
Yeah, I know. All right, later, Gator. Bye.